All right, if we could get you to take your seat, we'll move on with our next presentation for this evening.
That's fine.
That sounds terrible, but I'll give you a 10-minute.
All right, I know you haven't seen each other in a year, but you'll have plenty of time to socialize after this next presentation. So if we get you all to take your seats now, we're going to move on. Yeah, Rob, that means you too. All right, we're honored to have Dr. Matt Porteus as our next speaker this evening. Dr. Porteus is an associate professor of pediatrics at Lucile Packard Children's Hospital, Stanford, where he cares for pediatric patients undergoing stem cell transplantation. Dr. Porteus's research projects were the first to demonstrate that gene correction could be achieved in human cells at frequencies that were high enough to potentially cure patients. And he's a pioneer and founder of the field of genome editing, a field that now encompasses thousands of labs and several new companies throughout the world. His research program continues to focus on developing genome editing as a curative therapy for children with genetic diseases. Please help me welcome Dr. Porteus, who will discuss genome editing. How easy is it going to be to correct mutations that cause CF in patients? Dr. Porteus.
Thank you. All right, thank you very much for the invitation, and hopefully I'm going to hit the sweet spot between give enough science that you can understand where we are, but at a level, well, you're all really smart, so probably I don't even need to simplify it. But give you a sense of where we are in terms of actually trying to cure the disease. And I forgot the title I gave, so I made up a new title, which is Genome Editing, Opportunities and Challenges for Cystic Fibrosis, because I think that's really where we stand in 2016. I want to say, though, that I get to embarrass Dr. Wine, because when I was a medical student at Stanford, actually an MD PhD student at Stanford, I TA'd his class in neurophysiology as he was making the transition from understanding how channels worked in neurons to how channels work in cystic fibrosis. And so it's great to reconnect with old friends. Okay. So I'm an employee of Stanford, and they require me to disclose any potential conflicts of interest. And I've been fortunate enough to be involved as a founder, and I consult for a company called CRISPR Therapeutics, which is one of the few, there's about 4 or 5 companies now that have been founded around the idea of genome editing to cure genetic diseases. And I'll give a shout out that CRISPR has actually established a very close collaboration with Vertex Pharmaceuticals around using genome editing for cystic fibrosis. Now, what I'll be talking about is what's going on in my own research laboratory, but what we hope is that what I hope in my laboratory is that the academic work we do will have a pathway to patients through our connections with biotech companies. So I always like to start out with quotes, and for this talk, I went with this quote from K.R. Sridhar, who actually have no idea who he is. But what he wrote is a belief that finding innovative ways to make the world better is important. A mind in search of better ideas, even if they sound radical, is more likely to stumble across one. And I think this is a community that recognizes the importance of really both balancing feasibility and practicality with the goal to really take innovative and perhaps sometimes risky choices as we move forward. So no need to tell this audience, but monogenic diseases, so diseases that are caused by mutations in single genes, permeate all of medicine. And if you go to sort of the databases, there's estimated that there's probably over 6,000 such diseases in the world. And some of the recent efforts of sequencing random, whole cohorts, whole populations of people in Europe have suggested that actually 5% of us may actually have, quote, a monogenic disease, meaning that we have mutations in both copies of the gene. Now, most of the times, of course, that does not lead to any significant symptoms. But what I've listed here are a number of diseases in which they are severe, life-threatening, and for most of them have critical unmet medical needs. What I've put in green, of course, are disease. Sickle cell disease is actually the primary focus in my lab. I'll mention a little bit in my talk about our work around another genetic disease of the blood called severe combined immunodeficiency or bubble boy disease. And through my talk, I'm going to talk about how genome editing, both the opportunities, as I mentioned in the title, could impact on cystic fibrosis. But I'm going to end with talking about the challenges that we're going to need to solve in order to really translate this to patients. So the idea is that genome editing is actually a method to correct disease-causing variants. And I'm going to try to be really careful here not to call these things mutations. I think mutations have a pejorative context, and I don't think that we should be calling people's sequence mutations. I think we should be calling them variants. And in the end, the idea of reverting variants from something that might cause a disease to something that doesn't cause disease is the ultimate, I think, in personalized medicine, which is, of course, out there in the nomenclature. So what is genome editing? Well, while we think of it as reverting these disease-causing variants, in fact, it's a way of actually mutagenizing the genome, but in a very precise way. So we're going to take the sequence of DNA and we're going to change the sequence of DNA in a very controlled fashion, in a precise fashion. And the way this works is that we design specialized proteins that are designed to recognize a specific sequence in the entire genome, so the entire 3 billion base pairs, and they recognize one site, and at that site, they break the DNA into two. They create a double-stranded break. Now, normally, double-stranded breaks can be quite harmful to cells, and so cells have redundant ways of repairing that double-stranded break and fixing it. So one of the ways that cells repair that double-stranded break is through a process called non-homologous end joining, which simply involves stitching the two ends back together. And through the iterative cycle of break and repair, what can happen is through this NHEJ pathway is you can create little mutations at the site of the break. And so this is a really great way of breaking genes. But if we're talking about fixing genes, this really isn't what we want to do. Instead, we want to fix a mutation. But fortunately, the cells have another mechanism of repairing double-stranded breaks called homologous recombination. And in this mechanism of repair, the double-stranded break is created, and the cell, through a mechanism that in Silicon Valley is known as Apple C, Apple V, so a copy and paste mechanism, you make a copy of a piece of DNA that is nearly identical or absolutely identical to the region around the break. And what we do is we design this donor DNA so that it has just the sequence changes we want, and the cell will take those sequence changes and integrate them into the genome. And so this is a way that we can change specific bases, specific nucleotides in the DNA of a cell. Now, what I want to just highlight and what I'm going to go through in the next few slides is that we can change more than just a few bases. We can put in large stretches of DNA, we can put in new genes, we can put in whole cassettes of genes, and each of these might have implications about how you would apply it to one day curing cystic fibrosis. So there is sort of two basic ways of thinking about genome editing. One is that you can change the sequence that exists in one person to the sequence that exists in another person. And the reason I bring this up is over the last year or so, I've been had the opportunity to be in a lot of conversations with bioethicists who have been thinking a lot about what it means to change the sequence of DNA. And when I talk to them about whether you can change from the sequence of one person to another person, they don't really have many problems with that. So for cystic fibrosis, what that would mean is you would take a disease-causing revertant and change it back to a base or a sequence of bases that exists in someone else that doesn't cause this disease. But there's another approach to genome editing, which is take the DNA sequence and modify it in a way in which we create an entirely new sequence that has never been in a human being before. And the idea is by changing this is that by doing that, we control the way the cell works and we create a phenotype of the cell that is good. And so for cystic fibrosis, this might entail not simply reverting a pathologic variant back to a non-disease causing variant, but actually putting a fully functional copy of the cDNA, which is a copy of the gene, back into a precise location. And I'll talk about that in a second. The bioethicists who I've talked to get that this is probably okay as well, but then we're going to get at the end of my talk about where they have some problems with this. Now, if you read the scientific literature, what you read is that genome editing where you create mutations is much easier to do than genome editing where you fix or you revert specific changes. But what I want to show here is in fact, if you understand how to fix changes really well, we can actually get, for every mutation we create, we can actually fix 1.3. So we have a ratio of 1.3 to 1. But I think this is, I'm going to come back to this in a minute, is that in the process of genome editing, what you're doing is you're taking a sequence and you can either change it to something you want, but you're also occasionally going to get something you don't want. And I think that's something we need to really think about as we think about applying this technology to genome editing. So right now, there are 5 different classes of these nuclease proteins that can be engineered to recognize a specific sequence in the genome. They have been developed over the last 20 years or so. They have different characteristics, and they are in different stages of clinical development. Probably many of you have read about this new class of nuclease called the CRISPR-Cas9. And this class of nuclease has been incredibly exciting, not because it's doing anything particularly different than these other nucleases, but it's because they're really easy to make. And the word is, and it's true because I have high school students in my lab, is that they can come into the lab and instead of taking a very sophisticated graduate student months to years to make a specific nuclease, you can have a high school student come into the lab, take a recipe, make a CRISPR nuclease to a specific site in the genome, and in a couple weeks you have something that works really well. So what this has done is expanded the field of genome editing to an incredible array of labs, which was introduced earlier. And then in the last column, I just want to point out is that there are a number of companies, including the company I have a stake in, that have licensed the intellectual property around these nucleases in order to develop them for therapeutics. And in some ways we're competing, in other ways it's a situation of a rising tide floats all boats, because if they're successful, that helps us and vice versa. Okay, so, whoops, the bottom is cut out here. So I want to go through some of the different ways we can use genome editing to change the DNA sequence. So I mentioned the first thing is you make a break and then you create a deletion or a small deletion at the site of the break. And this is a really great way of inactivating a gene, so knocking out a gene or knocking out a genetic element. And as I think about how one might apply that to cystic fibrosis, I can't think of how it would help, and I actually worry that you could make things worse. So supposing somebody had an allele which didn't make enough of the protein, but made a little bit, you could actually make things worse by turning a hypomorph, so something with a decreased function, into something that had no function. And so I think we need to think really carefully about how to avoid this potential problem. But as I said, we really want to fix genes, and so that's going to mean that we have to harness this process of homologous recombination. And the first sort of simplest way of harnessing homologous recombination is to design a nuclease that's very close to the mutation or the variant that you want to correct, deliver a donor vector that will change that variant, and so you just have one corrected base. And obviously, the relevance to cystic fibrosis is great. And since this is a disease in which many patients have the same mutation or the same variant, it's possible one could design a system that attacks this delta F508. But this is also a disease in which patients have a large number of variants. And the number of variants is so large that I think it currently is not practical to think of developing a gene correction system for every single variant. So that leads to sort of the next concept, which is the idea of using genome editing not to change just a single base, but to allow us to make a break right where the cystic fibrosis gene starts, and then take the cDNA, the functional mRNA copy of the gene, and put it right where the start site is. So now the gene will be functionally fixed. And so I'm going to give an example of how we've used that for a different disease. So this is bubble boy disease. This is actually a series of pictures from a single patient, David Vetter. And one of the things, and I'm going to bring this up because I think it's a really important point, is that this is a disease which human biology has shown that we only need to fix one cell, one stem cell, and that stem cell can reconstitute the entire immune system. So this becomes something, well, if we only need, because we only need to correct one, it becomes something we might be able to think we might do quite easily. But like I mentioned, this is a disease where mutations are scattered throughout the genome. So there are certainly hot spots where there are lots of mutations, but you can see the disease is characterized by mutations throughout the genome. There are a few patients, and this is about 1 to 2% of patients, who have deletions of the entire gene. And so the strategy I'm going to describe won't work for these patients, but it will work for the 98% of other patients. And so the idea here is that we're going to design a nuclease right next to the initiation ATG start site. We're going to make a break, and we're going to insert a copy of the wild-type cDNA at that site. And so what we need to show is if we do that, do we actually rescue the function of cells? And so a little piece of data is that we've done this. We've taken our donor construct, our correction construct, and we've made T cells, which is where the important cell type for this mutation. And we've asked for the cells that we've introduced this cDNA versus the cells that are wild type that don't have any mutations, do they behave equivalently? And what we find is that they do behave equivalently. Now, what we'd have to do for cystic fibrosis is validate that when you put the cDNA right into that location, it behaves the same as a wild type gene. Now, it may not have to behave completely equal. It just has to work well enough to rescue the disease pathology that we see. Okay, so there's one. Now, another approach would be not to take the cystic fibrosis gene and put it back into the cystic fibrosis gene, but instead is to take a promoter, so a region of DNA that drives expression of a gene, connect it up to the cystic fibrosis gene, and put it into a safe place in the genome. So we define one place in the genome, and we say we're going to put this the cystic fibrosis gene in this new place, not where it's supposed to be, but in a new place that's safe. And so if for some reason these two approaches didn't work, this would be an alternative approach where we'd find one place and we'd put this cassette. Now the challenge with this is that the cystic fibrosis protein, the CFTR, is not expressed in all cell types, and so we wouldn't necessarily want it expressed in all cell types. And so figuring out how to only get it into the right cell types might be challenging, but it's possible. And then the final approach would be to take the cystic fibrosis gene and put it into some other gene that was expressed. And I can't really think for cystic fibrosis how this would be relevant. It's potentially relevant for other disease genes. So as we've thought about how we would turn genome editing as a concept into a real therapy that we would treat patients with, we've generated sort of a checklist of things that we need to be able to understand. The first thing is does this approach address an important unmet medical need? And I think this audience and the last talk have convinced me that, of course, we need better curative therapies for CF. We then have to decide whether we're going to use an homologous recombination-mediated approach versus an NHEJ-mediated approach. We have to figure out whether we can correct a gene defect or we're going to use synthetic biology where we create a new genome. Are we going to do this manipulation in cells that are outside the body, or are we going to have to do this inside the body, so ex vivo versus in vivo? What cell type do we need to modify? It's all well and good. I could probably correct the CF gene in skin cells, but that's not going to do us any good. So we have to get at the right cell to correct. we have to figure out what's the frequency we need in order to change the disease course. If I can only modify 0.1% of the cells, I doubt that's going to have an impact on the disease. But it's possible that, and you guys are going to teach me about this, or is maybe as few as 10% or 5% of the cells, if they were functional, could have enough of effect to change the disease course. But I think we need to understand, what is that frequency? What is the bar we need to get over? Once you sort of have these in place, then you start to get into the bioengineering aspects. Can we actually do that? Can we actually deliver our genome editing components to the cell type we want? If we're doing an ex vivo approach, do we actually know how to take those cells and transplant them? Can we make sure that we can evaluate this is actually going to work? And of course, we have to make sure that this is safe, and do we have mechanisms to evaluate whether it's safe? So I'm going to take a little detour now and talk about how we've applied some of these principles to a different genetic disease, and this is sickle cell disease or sickle cell anemia. Now, this is a disease that's caused by a single mutation in the beta globin gene, and it's a homozygous recessive disease, just like cystic fibrosis. And when you have sickle cell disease, when your red blood cells lose oxygen, they adopt this sickle-like shape, giving it the name of its disease. And what happens with these sickle red blood cells is they're very stiff, and so what they cause is blockages in our blood vessels, so our oxygen is unable to get to the tissues, causing an inability of oxygen getting to the tissues, and that can cause strokes, it can cause pneumonias, but the most frequent manifestation of sickle cell disease is recurrent bone crisis, as the bones don't get enough blood, and this is a drawing from a sickle cell patient sort of depicting the pain that these patients are in. And the survival curve for patients with sickle cell disease is the median survival, even in 2016, is somewhere in the mid-40s for patients with this disease. Okay, now I just want to go through, going off on this a little bit, but it turns out that where we look and see where the sickle cell variant is, it nearly completely overlaps with where we find malaria. And 50 years ago, actually, almost 60 years ago, Tony Allison, who actually lives in Mountain View right now, and other papers have shown that people who have one copy of the sickle gene, one non-sickle cell gene, are actually resistant to dying from malaria. And probably many of you know that the thought is that in cystic fibrosis, if you have one cystic fibrosis gene and one wild-type gene, when you are in an area that has a lot of cholera, it actually prevents you from cholera, although definitive study proof of that probably remains to be determined. So again, that's why I'm trying to be careful about not calling these mutations, because in the right context, they're actually beneficial. It's just that we happen to live in a world where there's no malaria and no cholera, which is wonderful. And so these variants no longer have any utility. Okay, so again, to summarize, if you have this sequence, amino acid sequence, you don't have sickle cell disease, but you have a higher risk of dying of severe malaria. If you're heterozygous, you have no sickle cell disease, but you have a lower risk of dying of severe malaria. If you do have sickle cell disease, you're actually even at higher risk of malaria. So this benefit counteracts this detriment. So for sickle cell disease, we're trying to fix the red blood cell, but the red blood cell only lives for about 120 days. So to actually permanently fix the disease, we actually have to go back and fix the stem cell of the blood. And so this concept that you can't just fix the mature cell of the lung, but you've got to figure out a way of fixing the stem cell of the lung in order to have a lifelong cure is, I think, one of the challenges that we have to face. And so what we know in sickle cell disease is that if we take the stem cells from the hematopoietic stem cells, the blood stem cells from somebody who doesn't have sickle cell disease, and we give them to somebody who do, that we can cure them of the disease. Now, this is a complicated process. Actually, this was first proven in 1984. And now we know that if you have a sibling who doesn't have sickle cell disease, who's an immune match to somebody who does have sickle cell disease, we can cure those patients about 95% of the time. Now, the problem is, for sickle cell disease, is that only 10 to 15% of patients actually have an appropriate donor. And so that leaves the remaining population out of luck. And when you have to do a bone marrow transplant from alternative donors, the outcome is much poorer. And by the way, this process costs about $300,000 to $500,000. So for sickle, so I talked about in SCID that we might only need to correct one cell. For sickle cell disease, how many stem cells do we need to correct? And without going into the details, there's evidence from stem cell transplant patients that we probably only need to correct somewhere between 2 to 10% of the blood stem cells in order to cure this disease. So that's our benchmark. That's where we need to get. Let's get to 10%. Can we get there? And so the idea then is not to use stem cells from someone else, but to take the patient's own stem cells, revert the sickle cell mutation, and then transplant those cells back into the patient and thereby cure them of the disease, and we need to hit that 10% level. So, as I said, we're going to use homologous recombination, we're going to correct a mutation, we're going to do this in cells outside the body, we're going to use the hematopoietic stem cells, and I put here 2%, but let's give ourselves a little buffer and try to get to 10%. So how are we going to do that? Now, I just want to point out that this idea of reverting the sickle cell variant is not a new idea. And in 1985, Oliver Smithies, who actually ended up winning a Nobel Prize for other science that he did, described the first time we could actually change that beta globin gene. Now, I want to point out this was in really the bad old days, because this is a Xerox copy of the paper, which you can tell sort of by the fading over here. And I had to put my name on it because I didn't want somebody to steal my Xerox copy, because that took a lot of work. And most importantly, I put a little check mark here, which meant I actually read the paper. I didn't simply Xerox the paper, I actually read it. Now, nowadays we download PDFs and it's impossible to tell whether you actually read it or not. And I probably have 20 copies of this paper on my computer because I can never find where I deposited the PDF of the other one. So, all right, so how are we going to go about doing genome editing for this disease? So we're going to first have to make an engineered nuclease that will make a specific break in the beta globin gene, and then we're going to have to give our correction, our donor DNA, to the hematopoietic stem cells. Now, what we learned, and I think, again, this is important when we think about cystic fibrosis, is if we just put DNA into cells, it turns out that cells think that that's an invading pathogen. They think this is a virus that's trying to infect me, and they react poorly to it. They say, this is an invasion, I'm going to activate an inflammatory response. So we have to figure out how to get these genome editing components, this genome editing machinery, into cells without triggering alarm bells in the cell we're trying to correct. So we do this in two different ways. So we isolate our hematopoietic stem cells, and we deliver our CRISPR-Cas9 nuclease by basically mixing the nuclease with our cells and running an electrical current. We zap them, it's called electroporation, and this floats into these cells. And so it turns out, it sounds sort of barbaric, but these cells tolerate that pretty well when we deliver this as a protein molecule, not as a DNA molecule. And then we have to deliver our donor vector, which has to be DNA. And so what we do is we package our donor into a virus called adeno-associated virus. And some of you may be familiar with this type of virus because it's been thought of for cystic fibrosis. And so this allows us to get the donor DNA into cells. And for reasons we don't understand, by delivering it to this AAV, it gets past the cell's sensing machinery, and we can get that DNA into the nucleus. And when we do that, what we find is that we can, using a cassette in which an AAV that would turn cells green if we get integration, what we find is in hematopoietic stem cells from different people, we can get on the order of 20 to 50% of the cells turning green. So we're getting high frequencies of homologous recombination by turning cells green, and they stay green over time. And if we take those green cells and we put them into special colonies, special media, we can see that they make all the different colonies that they should make, all the different blood cell colonies that they make. If we take them and we put them into mice and ask, can they make blood in an animal, not just in a tissue culture dish, we find that they can make blood in an animal. And if we take the stem cells from one mouse and then take the human stem cells from one mouse and put them into the next mouse, they can do that. So they have the properties of making all the lineages of the blood, and they have the properties of being a stem cell by being able to perpetuate themselves. So that's great. We can make them green. That's all cool. People can buy green fish now. But we need to change that one nucleotide, which is right here in the beta globin gene. So now instead of making a donor vector that's going to turn cells green, we're going to make a donor vector that switches that one mutation. And what we find is this is a little bit misleading, but in stem cells from patients with sickle cell disease, about 20% of the genes are unchanged. We didn't do anything. About 60% of the genes, we actually created a new mutation, and that goes back to this problem of how do we avoid making things worse. Now, in this case, the beta globin gene is about as bad as you can get, so knocking it out, making it worse, you can't make it any worse, but in about 20% of the cells, and actually in other samples, we can actually get to 40 to 50%, we actually can correct the mutation. So we think we're on our way, we're on the way to a process now where we think we can isolate stem cells from a patient. We can define how many cells we need to isolate. We will bring them into a cell processing facility, which Stanford University has now built. So what's called GMP, or good manufacturing practice, which means you basically got to wear gowns and hoods and you walk through doors and you sign in and out. And it's all very sterile. And we think we can make all these components and revert that mutation and then give those cells back to the patient. Now, what we have to do for the blood system is we have to get rid of all the other stem cells, create space for our corrected stem cells to move back in. So we do that by giving high doses of chemotherapy. And what we're hoping is, so we're in the stages now where we've proven this in the laboratory, and now we got to work our way through the FDA, and we got to and we're hoping we may be able to start a clinical trial for this in the early part of 2018. So that leads us to, we know how to engineer the cells, we've tested the red blood cell function, we've tested the stem cell function, and and the last sort of thing we have to do is use a combination of assays to make sure that we're not causing more harm in the process of doing this process. Now, I showed you that paper from 1985 from Oliver Smithies, and the frequency in that paper is he was getting 1 out of 1 million cells modified at the beta globin locus. And here we are now almost over 30 years later, where we finally exceeded this 10% threshold. We're now at 20% at the right gene in the right cell type. And so again, highlighting that good ideas sometimes take a long time to manifest themselves. Now, I sure hope it's not going to take till 2046 before we can be talking about where we are for cystic fibrosis, because I think we can learn from this and build on it and accelerate what it will take. So let's think about how we might do this for cystic fibrosis. So one idea to mimic what we do in the blood system is if we could isolate lung stem cells, and identifying what a lung stem cell is actually still quite a process, is then modify them ex vivo and then figure out how to transplant them back into a patient. The advantages of this is that we control everything in the laboratory. We control it in our Petri dish. We can easily measure how effective we are, and we can easily measure whether there was any off-target effects. What's the problems with this approach? Well, how do you isolate these cells? How do you modify these cells? How do you create and purify enough edited cells to be therapeutically relevant? And how do we transplant those cells back into the lungs? Now, do I think these are all answerable? I absolutely think they're answerable. Do I think they have easy answers to them? No, I don't think they're easy answers. So that would be schematized here, which is these are pictures of what we call bronchial epithelial cells. So we would use our CRISPR system and probably AAV to modify these cells and then somehow deliver them. And I'm sorry I chose such an ugly picture of a lung, because clearly this is too late. And that's going to be another challenge, is that we're going to want to do this in lungs before they get too sick. because once they're really sick, we probably can't reverse it. And that's going to really take some thought process with your community, the FDA, and scientists to say, when is it appropriate to use an experimental therapy, not when it's too late, but not when it's so early either. And there's going to be a sweet spot there, which I don't have the answer to, and it'll take a community to come up with an answer to that. Now, the other approach would be to do in vivo editing. And there, I'm going to show two slides about how you might think about doing this in vivo. So first of all would be to take the genome editing components, the machinery, and deliver them through a vein and hope that they migrate to the right tissues so that they get to the lungs, but they get to the intestines, they get to the pancreas, if you're male, I guess if they get to your seminiferous tubules, and that they revert that underlying, or they correct the CF gene in a way that makes it functional. So again, what I highlighted is this might be a way of edit cells throughout the body, including all the tissues that are affected by this disease. It means we don't have to manufacture the cells outside the body and give them. And in contrast to some of the other gene therapy approaches, since we're permanently changing the genome, this would potentially could be lifelong. You wouldn't have to redose. Now, what's the problems with this approach? Well, one problem is because we're doing it in the body, it makes it harder to monitor if we've been effective and safe. And then the question is how do we deliver this machinery to these tissues in an effective way? So for the donor vector, I think AAV is a reasonable way. We know that there are AAVs that get to all these tissues. The problem is we can't express the nuclease for prolonged periods of time, because while it's a really powerful protein, it's a foreign protein. And that means that if you have it expressed in the body, it's like a vaccine. And our immune system will see it as something foreign and will actually kill the cells, the same cells that we've tried to modify. So we have to figure out a way of delivering that nuclease in a way that, sort of in what we call a hit and run method. Make a break, go away, and be done. And so I think there are different ways of thinking about how you do that, whether it's through mRNA delivery, and I have colleagues that have looked at how you can deliver mRNA to the lungs through an IV route, or through maybe a nanoparticle that's not a virus. Now the other way, and probably many of you, well, all of you are familiar with the fact that your sons and daughters, or you, aerosolize. You breathe things in. The lung is an accessible organ through breathing. And so another potential way of delivering the genome editing machinery is to breathe in the genome editing machinery, or perhaps be even a little more invasive and have a doctor do a bronchoalveolar lavage and put a tube down and squirt it into the lungs so it gets delivered directly to the lungs. So the idea here is that, of course, the lungs are the major cause of disease, not to minimize the issues of the pancreas and the gut and the other organs, but certainly that's the most important. Again, it doesn't involve cell manufacturing, and again, it could be permanent. The problems are just like the intravenous, less ability to monitor efficacy and off-target effects. And really, I think the challenge with this approach, and this is what has plagued other gene therapy approaches, is how do you get your machinery through the thick mucus and the inflammation that's in these lungs? Can you figure out how to get it to the right cell type in an efficient enough fashion to create a frequency that's high enough that's going to change the course of the disease? And then we have the other issues of immunogenicity and genotoxicity. So I have a lot of question marks here. So I think we need to correct things. Whether we correct a mutation or we insert a cDNA depends. I think both. I think delta F508 is a common enough variant that causes CF that we should design a system that corrects that mutation, and maybe some of the other more common variants. But I think if we really want to approach every variant, then we have to think about a synthetic biology approach. Are we going to do this by manipulating cells outside the body and giving them, or are we going to do it by in vivo? I don't know. You know what? I think we got to do both. We got to try both. It's really important, I think, for us to figure out which cell type we need to modify. We need to figure out whether 2% or 10% or 50% is going to be necessary. Why? Because it sets a bar. It sets a bar for us to say, okay, that's what we need to hit. I think one of the problems, and I don't mean to bash my colleagues, is we will publish papers saying, 1%, that's fantastic because I only got, 1 in a million before, but maybe 1% is not enough. Or maybe 1% is fantastic, but we just don't know yet. And so figuring this out by doing mixing experiments, I think would be really important. And maybe there are people who know and I just don't know, so I want to be informed. And then how are we going to do this? What is the bioengineering strategy that we're going to do this? So I'm going to end with just a few thoughts on sort of the ethics of genome editing. This is a little bit more relevant for sickle cell disease, which is that we can design a very sophisticated process of making cells and giving them to cells, and I thank you for the kind words I've heard today about Lucile Packard Children's Hospital, and I think we can do this here. But for sickle cell disease, most of the patients live in this part of the world and this part of the world, which are not resource-rich parts of the world. So we got to figure out how to take this sophisticated process and get it to the millions of patients that actually have this disease. There's about 100,000 patients with sickle cell disease in the US and probably an equivalent number in Europe. The other thing that probably many of you have read about is this concept of doing germline genome editing or heritable genome editing. And the idea here is before somebody even has the disease, can we use genome editing to fix the mutation in the zygote or possibly even in a germline stem cell before you even have a zygote? So in a stem cell that would make sperm that would make the zygote, or an egg stem cell that would make an egg that would make the zygote. Now, when we've been in conversations about this, people have pointed out, well, you don't really need to do this because PGD, pre-implantation genetic diagnosis, and we just heard about it, could accomplish nearly everything genome editing would. But you guys are going to tell me more about this, but when you go online, what you learn is fortunately, there are now people who are growing up into adulthood who have CF and marrying people who have CF. And if they want to have a child who doesn't have CF, PGD is not going to work from them. So if this rare situation, perhaps we need to figure out how to do that. Now, are we crossing a Rubicon here that we don't want to cross by manipulating the DNA that will be passed down to future generations? Or is this a disease that's so terrible that it's worth crossing that Rubicon? And I have my internal opinions, but I'm going to keep them to myself about whether that's worth it or not. I just want to point out also that if somatic cell editing, so correcting the lungs, were really effective, you wouldn't actually need to do that. But it would lead to this paradoxical result, which is that there would, of course, be less need for heritable or germline editing, but you'd end up with more people who would pass along the CF allele to future generations. I'm okay with that, as long as we take care of patients so they don't go through the story that we just heard, which is awful. So anyway, I will end. I want to thank you for your attention and the opportunity to discuss what some of we've been doing. As many of you know, science is, what was the term? I alone will fix this problem. This is not an I alone will fix this problem. It takes a whole team of people, both in my own lab, my collaborators, our laboratory of cell and gene medicine, collaborators outside my lab, and a host of funding sources. It takes communities and support to do this. And we're going to cure cystic fibrosis, but it is not going to be a single person. It's going to be an entire community and an entire group of people doing this. And so I hope as our research progresses that we can become more involved in this. Stanford is a wonderful place to work. I will give a little shout out that one of the things we're trying to develop at Stanford is what we're calling the Center for Definitive and Curative Medicine, which is the idea is not to develop small molecules that help patients relieve, sort of dampen down the symptoms of the disease, but can we develop cell and gene-based therapies that actually cure patients of their disease? We don't have a website yet. We're trying to figure out how this business model will work. We're trying to figure out how we're going to, oh, there's my dog. We're trying to figure out how we're going to fund it between a mixture of foundations and grant support and partnerships with biotechnology, but I think we're going to come up with a plan. And I'd love to take any questions or comments. And again, I'd like to end a picture with my dog. Thank you very much. Yeah.
Okay, once again, we're going to open to questions. Again, please use the microphone and allow the volunteer to wipe it down after you're done.
Thanks.
Thanks for a very interesting talk. I was wondering if you could comment on whether the CRISPR-Cas9 or the Cas9 system actually represents an advance in technology that will make some of these pathways to therapies more likely to happen, because that's kind of what we've been hearing, and I didn't hear that in your talk.
Yeah. So it does and it doesn't. So the principle of genome editing did not depend on the discovery of CRISPR-Cas9, but the CRISPR-Cas9 technology allowed more people to enter the field. And so what we've done now is gone from a field where we might have had tens to hundreds of researchers to a field now where we have thousands, perhaps 10,000s of researchers. And as you probably guess, there's no doubt by bringing more people into the field, by allowing more people to access the technology through the CRISPR-Cas9 system, that we are accelerating the way to get cures for these diseases. So did I answer your question? I think yes, it did. It does, because it gives us many more people a way to attack the problem, whereas before there was a bottleneck for people to get into the field.
Okay, I can see that, but maybe being more specific,
Yeah.
I thought that the fact that you can target very precisely an area in the
Yeah.
genome where you're going to do the editing was the big advance with CRISPR over
Yeah.
homologous recombination, which just sort of happens to have to happen in the right place.
No, so the zinc finger nucleases and the tau effector nucleases did exactly the same thing. They created a break in a very specific location, but they were just nuclease platforms that were much harder to make. And so you had to be, my lab has been highly committed to those, but like I said, a high school student can make a highly active CRISPR-Cas9 in a couple weeks, whereas we've spent several years, for example, trying to make highly active ZFNs to the beta globin gene. So they are all really specific in making a break. It's just that the CRISPR allows you to get through that first step so much faster that you can move on to subsequent steps.
Thank you.
Thanks. Your analogy brings to mind it takes a village, and your village is quite large. Now, my question has to do with villages.
Yep.
The patent issues. How is the American and the global patent system either helping or hindering the work?
Yeah, so I think, boy, that's a hard question, right? So patents are good and bad, right? From the biotech and pharma standpoint, patent protection gives them the opportunity to invest large amounts of money knowing that they have protection. The downside is that patents can sometimes discourage multiple companies from working on it. In the academic world, I don't think any of us worry about that. We figure if I can show that I can cure 5 patients of cystic fibrosis by using genome editing, I'm going to let the patents play itself out, because there's going to be so much momentum, no one's going to get in the way of that. But you're right. We have Novartis, we have some big companies that are invested in this, and they are going to be a little slower in making investments in this technology unless they have patent protection. Now, the CRISPR, the CRISPR IP situation is still in flux. I'm going to put my academic hat on and say that while the two sides are battling it out and the lawyers are going to make a lot of money, I think what's going to end up happening is that the two sides eventually are going to come to an agreement. One side's going to realize, oh, I'm likely to lose, I better cut a deal before I officially lose, and I think it's all going to work out. And certainly the companies, the CRISPR-based companies, which is CRISPR Therapeutics, Editas, and Intellia, which then have partners with companies like Novartis and Bayer and GSK, are not acting in competition with each other. They all figure that, again, if we make something good, we'll figure out how to deal with the IP issues.
Okay, great.
Yeah.
Thank you.
A really fascinating talk, and I have multiple questions, but
Got to follow the rules, Jeff.
Come here, Rob, I'll give you a big kiss. So I think you might have covered this in the beginning, but a lot of us have heard the term gene therapy, and I heard a very funny talk by Eric Alton where he said gene therapy, wouldn't give it to my dog, gene editing cures everything. So I wonder if you could just succinctly for some of us, just contrast those two approaches.
I think sorry, I should have done that earlier. So there's some people, gene therapy among certain communities has gotten a bad rap. And when genome editing has been developed, there are people who said, well, let's keep genome editing as something separate, that this isn't gene therapy because we don't want to be tarred by this past reputation. I think that genome editing is a refinement of gene therapy. It's a more precise, a more controllable form of gene therapy. So in gene therapy, we were putting genes into cells, but we didn't always know, in fact, we often didn't know where they went. We would use a viral vector and they would they'd go somewhere in the cell, we wouldn't know where, or if they got into the DNA, we didn't know where they went. What genome editing allows us to do is control the change that we're making. It gives precision, which I think should give a degree of safety, certainly a degree of safety, but I think it also gives a degree of efficacy. So with classic gene therapy, which everyone sort of laughs at because there's all of 2 approved gene therapy products in the world right now, but we call it classic. One could never envision correcting the delta F508 mutation in the CF gene itself. Whereas genome editing, we can now actually think about doing that. And I was talking with somebody earlier. If you gave me a CF cell line in a dish and said, correct that mutation, we could probably do it in a month. That's in a dish. Whereas with other gene therapy techniques, you couldn't do that. Hopefully that answers sort of the distinction.
That's great, and that leads me to the second question, which was about so imagine you're in a situation that you discussed where you would have to be inhaling a vector. So although that vector ideally would precisely correct the variant, you still have the issue of which cells are going to take up that vector, and could you speak to that?
Yeah, it is. There's a mantra in the gene therapy world that the number 1 problem is delivery, and the number 2 problem is delivery, and the number 3 problem is delivery, and so on and so forth. And that's exactly right. How do we design our vectors either to hit only the cell we want, or if it happens to hit other cell types, is it a problem? I think when you think about an inhaled therapy, if there are certain cells that we particularly need to modify, but it turns out we modify other cells, I think we're okay with that, as long as we have the precision that we're not causing unwanted effects in those other cells that might cause problems. So I think hitting too many cells isn't necessarily too bad. As I said, my biggest concern is how do you get things to those cells in an efficient enough fashion? Because what I didn't show, and just for the aficionados, is that the frequency of that genome editing process is directly correlated to the amount of that donor vector you can get into the cell. So if you get very little of the donor vector into the cell, you have a low probability of correction. If you get lots of it into the cell, you have a higher probability. That's how we've gotten really good at it, is by getting that donor into the cell. So we have to figure out how to get a lot of that into the right cells in the lungs. Adam.
Thanks for the great talk, Matt. I like the comparison with SCID and CF. And one question is, when are you going to get involved with CF? I'd love to see this level of activity.
In fact, this coincidence is I was in my office today, a postdoc who's actually collaborating, working in doing lung organoids with an investigator at Stanford named Tushar Desai. We were in my office today talking about how we might start attacking CF. So we're thinking about it. And then actually So we're going to start working on it, maybe not tomorrow, maybe next month though. Yeah, so we're definitely but I think we're taking an approach that we don't think this is going to be easy. We're saying, look, if we sold that this next week we're going to have it done, I think we're misleading. I think I've tried to highlight, we think it's going to take a serious, thoughtful effort about how to do this, and that we want to make haste because there's too many patients that have this disease, but we want to do it in a way where we go one step forward, one step forward, one step forward, rather than one step forward, 2 steps back, one step forward, 2 steps back.
Yeah. Well, there's a lot to build on in the CF community in the area of gene therapy. One of the points that you made about nuclease-based approaches, I think one of the metrics that you showed was that you had 40% indels, I think, off-target effects.
Yeah. Well, those would be on-target effects
okay.
at the gene. So off-target, I didn't get into what Adam's talking about. By the way, Adam and I lived together for a year. So
many years ago
when I was in med school and you were doing CS. So there's the issue with nuclease-based editing is that we design the proteins to hit a very specific sequence, but at some probability they might make a break somewhere else in the genome. And do they cause a problem when they hit that other site? So that's what we call an off-target effect. What I was trying to refer to is that the indel at the site is an on-target activity, but might be deleterious because you could turn a low-functioning gene into a no-functioning gene and actually make those cells worse than they were before.
So is that a function of it being a double-stranded break that you're making as well?
Yeah.
So what about non-double-stranded break approaches to genome editing? Can you say something about?
So yeah, and I didn't put up a slide, but sometimes I show a slide where I show a belt, suspenders, and elastic waistband, and say there's 3 different ways of making sure your pants don't fall down. So I think that we need these competitive approaches where you develop a nuclease-based approach and non-nuclease-based approaches. In our hands, we find that the frequency that you're able to correct or modify a single base increases by 100 to 1,000-fold if you can make a break. Now, I know we're going to talk about, there are investigators at Yale who are using a different way of stimulating that process. The mechanism by which that works is much less clear. I didn't show a slide, but we understand sort of I can draw proteins and this is what happens and you get strand invasion and we can put everyone to sleep. The mechanism by these other approaches are a little less clear, so it's, does that mean as you translate and you don't understand how it's working, is all of a sudden are you going to go off a precipice? But I think it's fantastic that, we're competing. That's what's good for patients.
Great, thanks.
Yeah.
Again, Dr. Porteus. All right, well, this concludes our evening's program in the ballroom. We hope you've been informed and inspired. Please fill out your ORCID, by the way, that is purple, I've found out, evaluations for tonight's sessions. Rob's holding it up there, you can see the color. Well, that makes it easy. We now invite you to head over to the Blue Wave Lounge, where the incredible Beth Suffian will update us on the new Social Security medical criteria and what that means for those with CF seeking Social Security disability insurance. Suffian and Passamano LLP are hosting the reception, and CFRI is providing wine. The presentation begins in a few minutes, after which you can visit and peruse the raffle and auction items until 10 o'clock. Breakfast tomorrow morning from 7 to 8:30 a.m. here in the Grand Ballroom. Mealtimes seem to fly around here, and we'll start our morning program punctually, so please plan accordingly. Good night, and have a great evening.